Professor Alex Donald McLellan. He's a professor from Department of Microbiology and Immunology from University of Otago, New Zealand. And Professor Alex is an expert in cancer immunotherapy. Looking at his CV, he's already been working with a lot of them, including dendritic cell, NK cell, and recently in CAR T cell. And his research interest is focusing on CAR T cell against solid tumor, which we know is very, very difficult to tackle. So he have developing a lot of system in his laboratory, including dual CAR T using bidirectional promoters, inducible promoters, and tumor microenvironment sensor, and CAR T cell using metabolic boosting. But today he's going to share with us a very exciting, another exciting field of area in central control from within gene expression system for CAR NK and CAR T cell. So please welcome Professor Alex McLellan.
Thank you, Natia, for the kind invitation, and I congratulate you and your board on the foundation of the Talents Hub, and I look forward to being part of that. So I'll just share the screen. And I hope you can all...
Yeah, we can see the slide now, Alex. Thank you.
I can still hear you, so that's good, reassuring. So control from within gene expression systems, I'd like to focus on the tools that we've got available and that we're working with in New Zealand and the University of Otago in Dunedin. And really just a welcome photo to say that you're all welcome to come and visit. We currently have one student visiting at the moment I'll come to, but we are situated right at the bottom of the South Island of New Zealand in a beautiful old university setting that was there since the 1860s. And we're a long way from where you are, but we are connected because we are hosting a PhD student from Chulalongkorn University, Asmita Kania, who is also listening in to this Zoom presentation from our facility. So we're really pleased to be part of the hub, and I hope we can contribute. So I want to focus on what we have available in our laboratory setting that might be of use to the hub. This is our laboratory, and we've got quite a large space with facilities for lentivirus and Sleeping Beauty transduction of primary cells, and a number of postgraduate students will assist staff such as postdoctoral fellows and research assistants in the production of these cells. And the cells that we're generally working with are CAR T cells, or more recently CAR NK cells, and we've only recently managed to get a bioimager which has X-ray capabilities as well, since 2021. And we're currently using that at the moment for mouse experiments that are running. And we've had the NOD-SCID gamma immunodeficient mouse colony going for some time now, and that seems to be very successful. So what we do is we work with the classic lentiviral system, but also, like Chulalongkorn researchers have delved into Sleeping Beauty transposon system, because this does have some advantages of being able to increase the cargo size and perhaps better perceptions of safety because of the more random integration that the transposon will take inside the genome of the CAR T cell. So we get this working pretty well for lentivirus. This is the transduction frequency of a HER2 anti-solid tumor CAR, and we typically get quite high frequencies of CAR T cells transduced by that lentivirus. The lentivirus is usually produced by ultracentrifugation, and the titers are sufficient for a number of experiments, so we do that quite well. And one of the applications that we've looked at is the use of promoters as a single unit, but surprisingly to drive an additional gene on the reverse strand. So this was something we stumbled across some years ago, published in 2020, and it's just the setting up of a single promoter system to be able to drive 2 gene systems. And this could be quite useful for driving CD19 or CD20 CARs to fight antigen loss variants of leukemia or lymphoma cells. And the actual activity of these promoters in the reverse strand is strong. So you can see this setup experiment here. We've used the appropriate controls, one direction there, RFP in reverse, and GFP forward. And you can see there's very good activity in the forward direction, but also in the reverse direction. And some of these promoters you can trim down to quite basic elements, make them quite short. Some of these are only about 350 base pairs long, and you can have quite strong forward and reverse activity. So what that means is you can have 2 CARs expressed from one cassette, or you can have 1 CAR and a payload, which might be inflammatory cytokines or some other aspect that you might want to have expression of in a CAR T cell. So the current work using bidirectional promoters that we're undertaking is a comparison of different promoters. EF1 is a very well-known promoter, and it is also quite bidirectional. There are other promoters we've been investigating, such as the RPBSA, which I showed in the previous slide, and that is probably one of the more compact promoters. You can have a very small CAR cassette with just 1 promoter and easily achieve expression of 2 quite long polypeptides. So that's ongoing work at the moment. We're yet to report on the success of this approach, but it is a very promising approach to use a single promoter to drive the expression of 2 cassettes within the CAR T cell cassette. So one aspect we're also looking at is auto-inducible systems. So these are CAR T cells that only express genes when they come into contact with a tumor. And we're looking here at an activation-induced promoter. And this is an experiment which Josh Halpin, a former PhD student, now postdoctoral fellow, has been carrying out. And what we do is we do a lot of work in optimizing the design. You can see there are a lot of orientations here we've tried for expression of an inducible promoter driving RFP as a marker to see which system will give us the best titer or best expression. So you can see that the orientation is particularly important for obtaining titers. So this is a reverse cassette here with a poly A signal, and you've lost most of the production of the virus. The titers are very low, almost unusable. Some of these other orientations, forward orientation here of the activation promoter, or reverse orientation here of just this activation inducible promoter, gives fairly good titers. So it's really important when you're designing these to try a number of settings, and it's not always predictable which setting will work, unfortunately. So that was focusing on titers. If you look at the actual inducibility there, I've just focused on 2 of the better systems that gave good titers using an activation inducible system. So a CAR T cell comes along, hits the tumor, only then does the gene expression get induced. So this is in the absence of induction, so in the absence of tumor antigen, and this is with tumor antigen. You can see from the CD69 expression that most of the T cells here have become activated. And in particular, some settings here, reverse with a poly A signal, are quite inducible there. So you see most of the CAR T cells have shifted expression there and will express this RFP as a surrogate marker for an additional payload. The poly-A signal unfortunately drops the titers right off. You can see here by the inclusion here and one of the optimized settings there. So although it does improve for most donors the expression, it really is something to be avoided if you want to continue having high titers in your lentivirus system. It may be applicable for a sleeping beauty system where the poly-A signal might not interfere with the transposition frequency into the primary cells. So having selected really our favorite and optimized activation inducible promoter, you can also put in a setting where it's only driving one rather modest load, which is just the car itself. For the moment, we've left GFP in there as a marker. We also have a CMYC tag within the CAR, so we can tell how much CAR is expressed on the surface of the CAR T cell. And in this experiment, Sam Smith-Bell, who's a PhD student, has just tested the ability of the activation inducible promoter to protect the CAR T cells from forming too many effector cells at one time and possibly resulting in an exhaustion phenotype. So the setting there is similar to what Professor Terakura was looking at, which was iterative stimulation of CAR T cells, so hitting them with antigen, then hitting them again. And really that is the acid test for CAR T cells. They should not become too exhausted or form too many effector cells after the multiple stimulations. So if you look at the analysis here, EF1 is just constitutive promoter driving a CAR T cell. This is a NFAT NF-kappa B derived activation inducible promoter. And this is our own promoter that we're working on at the moment, activation inducible promoter, which is currently our favorite and currently being tested inside NSG mice. And you can see that after the first and second stimulation there, that with the EF1 constitutive expression of the CAR, there's a large population of effector cells forming, and this is increased after the second stimulation there. But if you look at some of the activation inducible promoter settings, NFAT, NF-kappa B, or our activation inducible promoter setting, you can see there's still a relatively large percent of naive or TSM cells, central memory cells with a slight stem cell phenotype, and a reduced number of effector cells. So we don't know what this means at the moment. It looks encouraging. I don't want to read too much into this figure, but as you say in English, the proof of the pudding will be putting these cells into the NSG mice with the self-driving CAR and looking at their activity against solid tumors. So they're currently being tested against MCF7, which we've derived to be HER2 positive. And you can see from this initial in vitro experiment there that the activation inducible promoter, as well as this other NFAT NF-kappa B inducible promoter, have surprisingly high cytolytic activity against the tumor, very similar to the constitutive promoter. This is another activation inducible promoter setting we've been tested, which has already been published by Guo et al. In 2022, and that's performing possibly not as well as some of the other inducible promoters, at least in our hands. And these are just untransduced controls or control empty lentivirus vector. So those are currently in the NSG mouse setting at the moment. We're testing those and we're about a week into the experiment, so we're very excited to see what's going to happen over the next weeks, and we're able to look at the results almost live with the bioimager visualization. So in parallel, we've also been looking at natural killer cells for immunotherapy, and we really just stumbled across an expansion protocol for natural killer cells when we were looking at ways to increase the proliferation of sleeping beauty transposed T cells. What we were finding was that the NK cells were overgrowing the cultures. And of course, we were initially disappointed until we decided that this was probably an opportunity for us and that it was probably a sign that we should study these natural killer cells. So natural killer cells do have some advantages over CAR T cells. And one of the features is that natural killer cells can respond to stress signals from natural ligands that might be induced upon cancer cells, and they can also be triggered by therapeutic antibodies if the antibody FC domain is ADCC intact. They can also be engineered with the CAR. This is usually with a lentivirus system, and we've had some success there, which I'll talk about in a minute. And the key to expanding these in vitro was not using CD28, CD3 beads as you can use for CAR T cells. There does not seem to be available a convenient bead system for expanding NK cells. So we use a cell line based system, an artificial antigen presenting cell system, which is using the MHC1 deficient K562 cell line. So we've engineered the K562 using Sleeping Beauty systems. We've put in 4-1BB ligand. We also have included CD86. There's some evidence that this can stimulate NK cells, and it also gives us the opportunity to use these same vectors for stimulating T cells. But really the most important components that we put into these artificial antigen presenting cells are the membrane-bound cytokines. IL-15 and particularly IL-21 is very important. The fact that they're membrane-bound appears to make their activity much more profound for expanding NK cells. We've also got available, although they're not always required for expanding cells, antigens that can be popped into these artificial antigen presenting cells that could be used for expanding T cells at the same time. So most of these constructs here, including an HLA disulfide trap peptide system, which is a single molecule, so a single polypeptide, including the peptide covalently linked into the HLA domain. These are all available from our AdGene collection should you wish to use these for your experiments. So the artificial antigen presenting cells, we've engineered these with a kit set, which is a transposon kit set. The advantage here is that you don't need to carry out lentivirus transduction, cell sorting, another lentivirus transduction, and cell sorting, and perhaps have issues of stability of the transgenes there. Each of these molecules that is now embedded intrinsically in here is linked to antibiotic selection. So we can ensure that the expression of these is maintained at a high level through antibiotic selection. If your artificial antigen presenting cell becomes contaminated, loses activity, or becomes senescent, you can start again. You just get these vector sets and start a very easy neon transfection to put the system back into your fresh K562 line. So the beauty of the Sleeping Beauty system is that you can get high expression of each of the co-stimulator or antigen molecules. I believe that this is better than lentivirus, and it's certainly better than transfections. We don't carry out transfections in the laboratory anymore. We simply use the Sleeping Beauty system because you get homogeneous and very strong expression of almost all transgenes that we've ever put into a range of cell lines. So we're just about to publish this in Immunology and Cell Biology. We've got a few tidy up experiments to do, and most of the, well, in fact, all of the molecules that you require for expanding NK cells can be found within the AdGene collection and will be described in that paper. So the key molecules there for expanding the NK cells was the discovery by others, in particular, that we've used, that you can have a membrane-bound IL-15 and it's much more potent than the soluble form. Likewise, IL-21 seems to be particularly good at expanding NK cells. Membrane-bound forms of those cytokines are particularly good at NK cell expansion. So that is the design there. There is the IL-21, a hinge region, and just the FC domain there leading to a transmembrane domain. You don't need to bother with the cytoplasmic domain. And that's just poking out quite high off the surface of the antigen presenting cell, providing stimulation for natural killer cells. So the nice expansion protocol there is optimized. We've tried a combination of cytokines and settled on IL-15 and IL-21. Both of those are membrane bound, and the membrane bound nature means they will expand to a much greater degree than soluble cytokines. As an example there, we've used 2 soluble cytokines we found were okay for expanding NK cells, but nowhere near as good as the membrane bound form of those cytokines. So a typical donor might have around 5 to 10% NK cells at the start. These are not activated, and we can enrich these up to 80 to 90% purity by the end. They also have retained their CD16 FC receptor, so we presume that these are ADCC competent and could synergize with therapeutic antibodies. So this is the expansion of NK cells. So even from a modest amount of blood, we don't need to bleed our donors dry, laboratory donors or kind people around the department that donate blood. Just a little bit of blood will give us a lot of NK cells, so we can generate from less than 50 mils, enough for mouse experiments. So very, very convenient mechanism for getting enough NK cells to work with. They're cytolytic, so this is the just a test of a couple of donors showing that they will lyse cells. We haven't extensively tested a range of tumor lines. We'd like to test some NK cell resistant lines, particularly with NK cells that have been engineered with a CAR. So having got our NK cells, we're particularly interested in asking how will they perform when you put a CAR in them. They've still got their natural cytotoxicity. Now they've got a CAR against solid tumors. They should be better at attacking solid tumors. So we put in a CAR, which is HER2. It has the CD28 domain, which Professor Terakura showed might be more adept at killing cells, and others have shown that it's more likely to kill solid tumors. So we didn't have too much trouble with the transduction frequencies, just using a VSVG setting, which is the standard setting for an envelope protein. But we are trialing other envelope proteins to try and increase the ability to get a higher number of transductants within our NK cell population. And one of those is a baboon envelope, which has previously been reported by others to give better transduction frequencies of NK cells, because the NK cells have more of the ligand for this particular envelope protein compared to T cells. That's why you might have to swap out the envelope protein. All our kit sets for artificial antigen presenting cells, antigens, HLA, and cytokines, as well as an optimized TETON system that we've taken. The TETON system is a tetracycline inducible system, made it even better. All of these are available from our AdGene collection. So if you have a look online, you can quickly search your way to Alexander McLellan AdGene and see that we've deposited most of the constructs there we've talked about today. So should you wish to make your own artificial antigen presenting cells, those constructs are all available on the public repository, which is AdGene. I'd like to thank the lab, and many of these have been in their time PhD students in the lab, and Josh Halpin now is carrying out some bioimager experiments for the NSG mice. He's currently a postdoctoral fellow. Ali Hosseini is a previous PhD student now at Kite Pharma, and contributed to some of the early experiments with the lentivirus and promoter selection. We also have within New Zealand, so if you visit us in Dunedin, you might also want to take a trip to Wellington to see the Mulligan Institute, where they're carrying out a CAR T-cell trial against B-cell lymphoma. And just across the ditch is Australia. And there we have two nice collaborators there, Mike and Phil of the Peter Mac Cancer Centre. And we collaborate extensively with those two and hold some joint grants. I'm really pleased to be part of the hub, the Thailand Hub of Talents, and Asmita is currently part of our team here as an exchange PhD student, and Natia and Fai have also helped with our progress here, and we're really pleased to continue with the collaboration with Chulalongkorn and the hub. I'm happy to take questions at this stage, and thank you for your attention.
Thank you so much, Alex, for an excellent and wonderful talk. So we are very excited to be able to work with you. Are there any questions from the floor? Oh, we have question from student, Alex.
Yep.
Hi, this might be a stupid question, but I just want to ask. We use the membrane-bound IL-21 and IL-15 for NK expansion. Do you still need to use like exogenous cytokines in maintaining NK CAR?
Yeah, surprisingly not.
So that would suffice in maintaining expansion of
Which
NK.
makes it nice because post-COVID, the transport of commercial goods into New Zealand is slow. Cytokines vary in their batches. And if you can have a well-established cell line in your lab, I think you're well ahead to have good quality control. And if everyone's using the same system around the world, that's even better. So different cytokines have different batches. They have different folding from different companies. One might be more active than the other. So it's nice to have that consistency. So a really nice question. And I'm glad you asked that, because you don't need any other additional cytokines other than the membrane-bound cytokines. Perhaps a little bit of IL-2 at the end can encourage some expansion, which is sometimes we do that, but generally it makes little difference even to have exogenous IL-2 and at the end at high levels.
Thank you so much.
Thanks.
So that's a great question, Kate. Thank you so much. Are there other brave students?
Hello, Professor McLellan. Thank you for your talk. My name is Tanyavi from Chulalongkorn University. I'm sorry if I didn't catch you, but the activation-induced promoter that will drive the expression of CAR, where does that activation comes from in the first place? Thank you.
That particular promoter is, it will respond to tumor antigen recognition by the CAR.
Oh, okay. So, but does the CAR comes first, or does the transcription by the AIP comes first? Which one is the chicken and which one is the egg?
Usually those promoters are a little bit leaky. So there's just a smidgen on the surface, and it's enough to generate a positive feedback loop.
I see.
So if they weren't leaky,
got it.
I don't think you get any activation, but they're just a little bit leaky. We try to make them we try to make the leakiness as little as possible, so that we try to make them as tight as possible. But even the tighter ones seem to have really good inducibility. So it's not like a Teton system in bacteria. You can switch it on and off. There's certain leakiness there.
Okay. Hi, good morning. My name is Chachanan, also from Chulalongkorn University. Thank you so much for your talk. And actually, I have a follow-up question from that about the AIP. So you said that the promoter can be a little bit leaky, so that, and then the feedback loop has to come from that first CAR molecule, right? That's expressed from the leakiness of that promoter. And then I'm wondering, like, once the cell has come into the vicinity of the tumor, and then the feedback loop will go on and on. So how is that promoter be different than the constitution additional promoter like, once it's already in the tumor vicinity?
Yeah, so the activation of that particular promoter is transient. And even with EF1 or a strong promoter, you'll find that CAR, as well as T cell receptor components, will internalize, and so you lose expression. So if you look back perhaps to Antonio Lanzavecchia's papers on T cell receptors, you see that T cell receptors are downregulated upon antigen triggering. And the same thing happens with the CAR. And there's been more work done from perhaps you could check out Michelle Sadelaine's papers showing that CARs are downregulated upon antigen contact. It may happen that with the activation-induced promoters, this is more profound. But even EF1 or some so-called constitutive promoters, the CAR will downregulate. We think we're getting less exhaustion, and we're certainly getting less effector cell production on the first antigen contact. So that might be providing an advantage, but it will affect this. I think it will affect the cycling of the TCR because it will be less or CAR. It will be less in the first place, and it may be on a tighter cycle. Great question. Thanks.
So much. Thank you very much.
Okay, so one more question from the Zoom that Dr. Nawapat, she is from Cancer Science Institute of Singapore. Her question is, did you check the cell exhaustion after you constructed CAR?
Did we check exhaustion?
Yeah, right.
Not yet. Yeah. We have the panel and we're going to do that soon.
Okay, so I think it's time for coffee break. Thank you so much, Alex. I hope you were here with us because we have a very good coffee break. But we're going to meet you in November. So thank you so much. Let's have a big round of applause for Professor Alex. Okay, Alex, thank you. So we're gonna have a 20 minutes coffee break and we're gonna come back at around 10:20.